Hello, everyone. I hope you're doing great today. It is a fabulous day to talk about the digestive system yet again. And so what we're going to cover in this lecture is we're going to talk about the accessory organs that secrete things into the gastrointestinal tract. Specifically, we're going to talk about the pancreas and its exocrine functions, its endocrine functions. We're going to talk about the magical organ, the liver. When in doubt on your HESI or any other test, if you have no idea what organ does something, it's probably the liver. It has so many different functions, so we're going to talk about the functions related to digestion and its other magical traits, and then I'm going to cut this lecture off, and in the next lecture we'll talk about chemical digestion and regulation of digestion. So that is where we are going. Okay, so we had talked our way through the gastrointestinal tract, and just as a reminder, we ingest food into the oral cavity, we masticate, we chew it, we form a bolus, then through deglutition we swallow the food and it goes back to the oral pharynx, and then we have this involuntary reflex. The food goes from the pharynx to the esophagus to the stomach, and then the distal end of the stomach is the pyloric sphincter, which is the gatekeeper. And so the pyloric sphincter lets chyme out from the stomach at 3 mL squirts, it's about 3 per minute, into the duodenum. So this picture is showing you the duodenum. That's just the proximal part of the small intestine, sort of makes a C, and you can find the pancreas. The head of the pancreas is sort of nestled into that C shape of the duodenum. And so I'm calling it the great intersection. So a lot of magic's going to happen in the duodenum because we have 3 things coming in. We've got that acidic chyme from the stomach. We're going to add the bile made by the liver but released from the gallbladder. And then we're going to talk about pancreatic juice and what's in there. So we have a lot of things. We got a lot of chemistry going on, a lot of the chemical digestion, and a lot of absorption is going to occur in the duodenum. So let's look at the pancreas first. And so we look at the macro anatomy or the gross anatomy. And so you can see they call this the head. And like I said, that's kind of the easiest way to find the pancreas. The pancreas actually in dissection is kind of difficult to find. It's kind of a mushy, non-colored organ. So this is the easiest way to find the head. It's nestled in the duodenum. This part is called the body, and this part is called the tail. And so this tail sort of goes posteriorly, and it's going to be retro behind the peritoneum. If you look at the microanatomy, it gives you some insight into its function. It has both an exocrine function and an endocrine function. And so looking here, you can see we've got the exocrine glands. So remember, exocrine glands secrete things out onto a surface. And so pancreatic juice is going to come from these epithelial cells. We're going to get some digestive enzymes and some bicarbonate. And then when you look at it microscopically, then too, there were these little islands. They used to call them the islets of Langerhans, if you remember Langerhans cells from skin. But a better name for them are the pancreatic islets. And so this is going to be their endocrine function, and they're going to make insulin and glucagon. So I'm going to talk a little more about that. So let's talk about the pancreas. So we're first going to cover the exocrine function of it, and that's going to be carried out by the acinar cells. And so when you have this little sac-like structure, it's called an acinus. And so these cells, they are going to make and release digestive enzymes for digestion. And they're also going to secrete a bicarbonate to neutralize the acidity of the chyme coming from the stomach. So the duodenum is really the one place in your body where there is an alkaline pH, and so a pH of about 8. So it is the only place, because we are acid makers. So these acinar cells, the enzymes that they're going to make amylase, which digests starch. They're going to make lipases, which digest fats. They're going to make proteases, which digest protein, nucleases, which digest DNA, and then bicarbonate, like I said, is going to neutralize that acidic chyme coming in from the stomach. Okay, so just like the stomach, if you remember, we've got those chief cells that had to make a protein digesting enzyme. And so pepsinogen is released in the stomach, and then the hydrochloric acid from the parietal cells activated. So the pancreas had the same sort of tough job. It has to make protein digesting enzymes, which could actually digest the cells themselves. So they're going to release those protein digesting enzymes in an inactive form. So on the cells that are on the sides or the walls of the mucosa of the duodenum, they are going to make enteropeptidase. And so that is the enzyme that can then activate those protein digesting enzymes. And so the pancreas releases trypsinogen, and then the enteropeptidase then activates that to trypsin. And then we have chymotrypsinogen, that's going to be an inactive form, and trypsin actually then can activate chymotrypsin. Procarboxypeptidase is an inactive form because it has pro in the beginning. Trypsin can also then activate that to carboxypeptidase. And so what all of these enzymes are going to do are they're going to take those short chains of amino acids, those peptides, coming from the stomach, and then they are going to break those down into small chains, so even smaller chains. So they just break bonds between amino acids. So that's why they're called peptidases. Okay, so this is a slide of the pancreas, and so when they looked at it, all they could see is that, well, there's some really darkly staining cells and there's some pale cells. And so the darkly staining cells, they call them the alpha cells, and then the pale cells, they call them the beta cells. So when things look different visually, they are going to have a different function. So remember, it's always structure and function. And so what they found later is that those alpha cells that stain so darkly, they actually produce the hormone glucagon. The beta cells that stain more pale, they actually produce insulin. So the pancreas is going to play the vital role in controlling our blood glucose levels. Okay, so how does that work? So when we've eaten and we start to absorb our food, the food is going to go into the bloodstream and blood glucose is going to go up. And so the pancreas has chemoreceptors for glucose, and also after a meal, they are going to release insulin. So after a meal, we're sort of glucose rich, if you will, and so we need to store that for in between meals or when we need more energy. And so the primary target is going to be the liver and the magical organ, the liver. And so what the liver is going to do is going to go through glycogenesis. It is going to build a very large storage carbohydrate molecule called glycogen. And so it's the liver's job, and also the skeletal muscles, that they're going to take up this glucose and store it for later. And so the liver stores about 12 to 24 hours supply. To be honest, I don't know the exact time. Different books, different references say different amount of time, but they're going to store glycogen for in-between meals. What happens when we're fasting or exercising, the pancreas is going to detect a drop in blood glucose levels, and then it releases glucagon because the glucose is gone. The primary target is going to be the liver, and then the liver can go through a process called glycogenolysis, and so it's telling you what it is. So we're going to lyse glycogen, and then the liver will release, it'll break off these little glucose units and release them into the bloodstream. And so the pancreas and the liver are going to work together to regulate blood glucose levels to about 0.1%. And so I'm going to ask you, so glycogenolysis, we've talked about this before. Can you remember what the stress hormone is that raises blood glucose levels? Okay, so here's just a pictorial representation of what we just said. So after a meal, blood glucose goes up, and then the pancreas releases insulin. The primary targets are going to be the liver, skeletal muscle, and adipocytes. So the liver is going to store the glucose in the form of glycogen. Skeletal muscles can also store glycogen. They don't share it with any other organs, but they're just going to store it for themselves for when we exercise. And then if there's a surplus, then it's going to be converted to fat, and we store our fat in adipose tissue. And so that, of course, is going to bring the glucose levels back down to a normal level. So the process insulin stimulates is glycogenesis, and that term genesis means creation of. I know it's a lot of glycos. You have to keep them straight. You can do it. Okay, so what happens in a healthy person is that their blood glucose after a meal in response to insulin is going to be maintained in homeostasis level. If someone has diabetes, and so this is showing type 2 diabetes, In this case, the pancreas works, the pancreas makes insulin, but the body becomes insulin resistant. So the organs, the liver and the skeletal muscles become unresponsive. So blood glucose levels are high. And so tying it to urinary, if you remember, if anything in the blood exceeds that transport maximum or that renal plasma threshold, the kidneys aren't able to absorb it, and so it's going to be excreted in urine. And so that's why that urine analysis, if you detect glucose in the urine, then that person may have diabetes. We talked about the non-pathological reasons that sometimes people have glucose in their urine. Perhaps they ate a whole bowl of Halloween candy. But just kind of reminding you about the I Love Lucy. Hopefully you watched it and enjoyed it, but that's why you end up with glucose in the urine. Okay, so what happens in between meals? So when we're fasting and also when we're exercising, then the pancreas is going to release glucagon. So when the glucose is gone, and so that's how I always remembered it. The targets are going to be adipose tissue, so we're going to get lipolysis. That's the signal for the fat to break down. And then what the liver is going to do, the liver is going to split off that stored glucose, so split it off from glycogen, so we get glycogenolysis. And then the other magical thing that the liver can do, which we'll talk more about in nutrition, is a process called gluconeogenesis. All that lactate that's made by muscles under anaerobic conditions, the liver can then take that lactate and then convert it into glucose. Also, the liver can build glucose from non-carbohydrate sources. So that's what that word means. So gluco for glucose, neo means new, and genesis means the creation of. So gluconeogenesis is when the liver in a fasting state or when we're exercising, when blood glucose levels drop, the liver is going to make sure that our glucose levels stay constant. And so it can split off stored glucose from glycogenolysis, or it can make glucose from non-carbohydrate sources, gluconeogenesis. Okay, so this is just kind of the verbiage of what I said. So that's for you. That's also for you. That's the verbiage of just what I said. One point that's very good, that's very convenient or fortuitous when someone has diabetes is that their liver, the kidney, and the brain, they do not require insulin. And so you still have brain function, you still have liver function, you still have kidney function. So vital organs are still going to function in diabetes, but that's a good thing. Okay. So this will definitely be on your exam. You need to know your glycos and you need to keep them straight. So this is the money slide. So this is everything I just said. If you take the word apart and look at it, it's telling you what it does. And that's one of the reasons I love teaching science. It makes sense. If you just kind of slow down and learn those root words, it's going to make your life so much easier. So you can either try to memorize it or you just kind of look at the word and understand it. So glycolysis, that converts glucose to pyruvic acid. We're going to talk about that process in the next week or the next chapter. And so that is the splitting of a 6-carbon molecule glucose to 2 3-carbon pyruvic acid. So that's where it gets its name. Glycogenesis is that formation, that creation of glycogen from glucose. Glycogenolysis is the lysis of glycogen. So now we are going to release glucose. We're going to break that off from the big, huge glycogen molecule that's going to be stored in liver and skeletal muscle cells. And then gluconeogenesis is when the magical organ, the liver, can take non-carbohydrate sources and make glucose. That would be a good matching question, wouldn't it? Okay, so now we're to the magical organ, the liver, one of my favorite organs. I know I say that about a lot of them. Just whatever organ I'm talking about, that's going to be my favorite. Okay, so when we look at the gross anatomy of the liver, it's pretty obvious that it's divided in half by this falciform ligament, and so that's that ventral mesentery. And so we get a right lobe and we get a left lobe. They act independently. Like I said, they are connected by this ligament. So if we look at this inferior view now we're looking at the underside. We're going to see the gallbladder there. And then we have this little magical area that's called the porta hepatis. So right there, let's zero in on that. And so we have 2 ins and 1 out is how I always remembered it. So what we have going in, we have the hepatic artery, and we know arteries are going away from the heart, and this is bringing that oxygen-rich blood from the left side of the heart. We have the hepatic portal vein, and I realize it's been a while since we studied the circulatory system, but it is bringing that nutrient-rich blood from the intestines. So we're getting the venous drainage through those mesenteric veins. And then we have an outflow, the common hepatic duct. One of the digestive functions of the liver, it's going to make bile. So we have 2 ins and 2 outs, and it's all happening at the underside, the inferior part of the liver. The hepatic veins are actually going to exit out the superior part. And like I said, you also see the gallbladder under there, and we'll talk about the gallbladder in a moment. So this just kind of reminds you of the circulatory system. And so what's happening, what's going into that hepatic portal vein, we're getting all this venous drainage from the superior mesenteric vein, the inferior mesenteric vein, from the splenic vein, from the gastric veins. It's all going into the liver. And so the liver is going to filter, detoxify that blood, and then also it has a major role in our nutrient balance of amino acids, and we just talked about its role in regulating blood glucose levels. All of these veins now, if you remember when we studied mesentery, is going to be encased in that double membrane fold of mesentery proper. Just kind of connecting all those dots for you. All right, so let's watch about the magical organ, the liver.
The liver is the largest organ in the body. Liver tissue is composed of a compact mass of multi-sided units known as hepatic lobules. Each lobule consists of a central vein, which acts as a tributary of the hepatic vein and conducts processed blood away from the lobule, surrounded by plates of liver cells. The liver receives blood from 2 sources. 80%, which carries digested food materials, arrives from the intestine via the portal vein and venules. The remaining 20% is oxygenated blood from the heart, which enters through the hepatic artery and arterioles. An exchange of materials takes place between the liver cells and the blood, which then passes into the central veins and returns to the general body circulation via the hepatic vein. Sinusoids, spaces between plates of liver cells composed of tributaries of the hepatic artery and portal vein, conduct the blood flow to the central vein. The bile caniculus carries bile juice from the liver to branches of the bile duct, which convey bile from the lobules to the gallbladder. When an adult is at rest, about 2.5 pints of blood flow through the liver each minute.
Okay, so the functional unit of the liver are the hepatic lobules. Doesn't it sound better if you say it with an English accent? They actually did a study and realized that people automatically assume that people that have a British accent are more intelligent. But we call them lobules, but you can call them lobules if you want to. But this is a representation of a lobule, and it's a hexagonal shape. And so at each corner of the hexagon has 6 sides, there is what's called a portal triad. And so that's what I was talking about. You've got 2 ins and 1 out flowing not only at the hepatoportus, but also on the triads. And so we've got the oxygen-rich blood coming in. It's going to be a branch off the hepatic artery. We've got this branch that's coming from the hepatic portal vein, so the portal venule, and then we have bile flowing out. So as blood is flowing through, it's being filtered, it's being detoxified, damaged and old red blood cells are going to be removed, and all the while these hepatocytes are also making bile, and the bile flows out these bile ducts and is going to be collected in the hepatic ducts. Also in the liver, so we studied those sinusoidal capillaries. So these are very open and leaky capillaries. In the liver are going to be resident macrophages and dendritic cells, and a guy named Kupfer saw them, so he got to name them after himself, but they're also called just hepatic macrophages. They are going to be removing bacteria from the blood as the blood is being filtered. As far as the digestive functions, The liver is going to make bile, and bile is mostly water, like everything else. It also has what are called bile salts, and these are going to be cholesterol derivatives. It's going to have some phospholipids, and what the chemical composition of bile is going to be these amphipathic molecules. And what that means is amphi, like both, it's going to have a polar end and a non-polar end. And what that's going to do is it's going to orient itself in a way that is going to take a large fat droplet and turn it into smaller fat droplets. And so this process is called emulsification. What emulsification does is it allows, we need small droplets so we have large surface area so the pancreatic lipases can get to all the fat and digest it. And so it's necessary for digestion. The bile itself isn't digesting anything, but it is going to emulsify the fat so the pancreatic lipases can work. Other things that are in it are bile pigments, the breakdown products of hemoglobin. We have water, we have electrolytes, kind of already said about the water. And like I said, it's going to be those bile salts that are going to emulsify those fats. What bile does is it enhances the absorption of fatty acids, cholesterol, our fat-soluble vitamins, which are A, D, E, and K. Most of that absorption is going to occur in the ileum because it takes kind of a long time to digest fats. In order to absorb fat-soluble vitamins, it works a whole lot better if you've had a meal. So if you are taking vitamin supplements, you should probably take it with a meal that they have some fat in that meal, and you'll get better absorption. But the liver's job as far as digestion is to make and secrete bile, which emulsifies fats. Okay, so bile does not digest fats. The pancreatic lipases digest fat. What bile does, it actually emulsifies fat. It turns it into these little micelles. It basically takes a big fat blob and turns it into a little fat blob. So while the liver is making bile, the right lobe and the left lobe are both acting independently, and then the bile is going to flow down the right hepatic duct and the left hepatic duct, and then you've got the common hepatic duct, and then it flows into the common bile duct, and then there's a sphincter here called the hepatopancreatic sphincter. And when you're not eating, this sphincter is closed. And so what's going to happen is this bile is going to back up into this little muscular sac called the gallbladder. While it's in the gallbladder, water is going to be absorbed. We're going to concentrate it about tenfold. And what's going to happen is when we do eat, particularly eat a fatty meal, there's going to be a release of a hormone called cholecystokinin. And that is going to cause this gallbladder to contract, this hepatopancreatic sphincter to open, and then we get this nice concentrated bile into the duodenum to help us emulsify our fats so then the pancreatic lipases can digest them. A common problem is gallstones.
The gallbladder is a small sac located underneath the liver. The gallbladder serves to store and concentrate bile. Bile is a yellowish-green fluid secreted by the liver and contains bile acids which aid in fat digestion and absorption. Bile flows through the bile duct into the duodenum, the first part of the small intestine. After filling the bile duct, it overflows into the gallbladder, where it is stored for later use. After a high-fat meal, the gallbladder contracts to pump bile into the duodenum. Gallstones are hard masses formed in the gallbladder. Gallstones may cause obstruction of the cystic duct and excruciating pain when the gallbladder contracts. This usually happens after a fatty meal and is commonly referred to as gallbladder attack. Blockage of the cystic duct is a common complication caused by gallstones. Other less common but more serious problems occur when gallstones become lodged down the path of the biliary tree. When gallstones block the common bile duct, they prevent bile from reaching the intestine. This causes jaundice, poor fat digestion, and subsequently leads to infection of the bile duct or cholangitis. Gallstones may also obstruct the pancreatic duct, forcing pancreatic enzymes to back up in the pancreas. This damages the pancreatic tissues and triggers inflammatory response. This condition is known as acute pancreatitis, or sudden inflammation of the pancreas. The most common treatment for gallstones is the surgical removal of the gallbladder, or cholecystectomy. Laparoscopic cholecystectomy is currently the standard procedure for gallbladder removal. This minimally invasive procedure requires only several small incisions in the abdomen, and thus results in less pain and quicker recovery. The cystic duct and cystic artery are clipped with tiny titanium clips and cut. The gallbladder is then dissected and removed through one of the incisions. After surgery, bile enters the intestine without being concentrated in the gallbladder and may not be sufficient after a high-fat meal. A low-fat diet is therefore recommended after removal of the gallbladder.
Okay, so gallstones are pretty common. They are more likely to occur in women and the elderly. Doesn't mean men can't get them. Men do get them. When someone has a gallstone, often the symptom is going to be pain in that upper right quadrant, usually after they've eaten a meal. As the food enters the duodenum, if they have gallstones, that cholecystokinin is released, that gallbladder is going to contract, and that's what's causing the pain, is that pressure. Increased risks, sometimes they run in families, but obesity, diabetes, also there's a higher correlation, women on birth control pills. Like I said, treatment is going to be that removal. Contraindications, meaning things you're going to advise against post-surgery to your patients, is to have them eat a low-fat diet because they're not getting that concentrated bile. That's the job of the gallbladder. It concentrates and stores bile. So this is a student very kindly gave this to me during the semester. She had her gallbladder removed, and so it's just showing you just exactly what we saw on the YouTube. The gallbladder is about 3 to 4 inches long, and it's attached to the liver by the visceral peritoneum, but they're going to clip it, and then they're going to cut it. And these were her gallstones. So, like I said, these are precipitated out, crystallized cholesterol from bile. And the common thing that it's going to block is that cystic duct. Okay, so our last organ in this section is we're going to talk about the magical organ, the liver, and this is a great TED Talk. It's one of my faves.
There's a factory inside you that weighs about 1.4 kilograms and runs for 24 hours a day. This is your liver, the heaviest organ in your body, and one of the most crucial. This industrious structure simultaneously acts as a storehouse, a manufacturing hub, and a processing plant. And each of these functions involves so many important subtasks that without the liver, our bodies would simply stop working. One of the liver's main functions is to filter the body's blood, which it receives in regular shipments from 2 sources. The hepatic artery delivers blood from the heart, while the hepatic portal vein brings it from the intestine. This double delivery fills the liver with nutrients that it then sorts, processes, and stores with the help of thousands of tiny internal processing plants known as lobules. Both blood flows also deliver the oxygen that the liver needs to function. The blood that is received from the intestine contains carbohydrates, fats, and vitamins and other nutrients dissolved in it from the food you've consumed. These must be processed in different ways. In the case of carbohydrates, the liver breaks them down and converts them into sugars for the body to use as energy when the filtered blood is sent back out. Sometimes the body has leftovers of nutrients that it doesn't immediately require. When that happens, the liver holds some back and stacks them in its storage facility. This facility works like a pantry for future cases when the body might be in need of nutrients. But the blood flowing into the liver isn't always full of good things. It also contains toxins and byproducts that the body can't use, and the liver monitors these strictly. When it spots a useless or toxic substance, it either converts it into a product that can't hurt the body, or isolates it and whisks it away, channeling it through the kidneys and intestine to be excreted. Of course, we wouldn't consider the liver a factory if it didn't also manufacture things. This organ makes everything from various blood plasma proteins that transport fatty acids and help form blood clots, to the cholesterol that helps the body create hormones. It also makes vitamin D and substances that help digestion. But one of its most vital products is bile. Like an eco-friendly treatment plant, the liver uses cells called hepatocytes to convert toxic waste products into this bitter greenish liquid. As it's produced, bile is funneled into a small container below the liver, called the gallbladder, before being trickled into the intestine to help break down fats, destroy microbes, and neutralize extra stomach acid. Bile also helps carry other toxins and byproducts from the liver out of the body. So as you can see, the liver is an extremely efficient industrial site, performing multiple tasks that support each other. But such a complex system needs to be kept running smoothly by keeping it healthy and not overloading it with more toxins than it can handle. This is one factory we simply can't afford to shut down.
Yes, so that's kind of why I call it the magical organ, the liver. You have a table in your notes, but let's kind of talk through some of those. So those hepatocytes are pretty amazing. They can carry out more than 500 metabolic functions. And as it's filtering the blood and it's receiving that nutrient-rich blood, it's going to basically process every class of nutrient. It plays a role in carbohydrate metabolism, lipid metabolism, protein metabolism, and just like that YouTube, it's also going to store things for us. It's going to regulate those levels, like I said, serve as a pantry and regulate plasma levels of our nutrients. The hepatocytes, they're so important. They have this amazing job. The fabulous thing is they can regenerate. So they are highly mitotic. They can regenerate themselves, even if there's only 25 to 30% of a liver left. We do not have a backup for the liver. If someone has kidney or renal disease, they can go through dialysis. There is not one for liver. So if someone has disease of the liver, cancer, hepatitis, that term means just inflammation of the liver. Usually that's caused by a virus or through damage through alcoholism or drugs. Cirrhosis of the liver, that is that last stage. That is when the hepatocytes basically, they just give up. They can no longer regenerate. It's going to be replaced by scar tissue, and so there's no liver function. So often if someone is on medication, long-term medication, then there's always going to be that blood panel. They're going to be looking for liver enzymes. They're also going to be looking for what's called BUN, blood urea nitrogen. They're going to be checking kidney function. So remember, we have like that one in, we can eat food, we can drink liquids. Our only outs are going to be through urination and defecation, and so the liver is going to help get rid of toxins and put it in the bile, which then is going to be excreted with the feces. So it's just kind of a really nice thing that the liver does for us. And so that word detoxification, it's a fancy word. People are, oh, I'm detoxing right now. This is kind of a nice picture. It basically, what that means is you take a harmful substance, the liver is going to use its many enzymes and convert that into a less harmful form, and then of course try to get rid of it in bile if it can. And so that's that term, detoxification. Other things, don't forget, it's filtering blood. There's those Kupffer cells. Also, when someone has a damaged spleen and the spleen has to be removed, remember the spleen's job was to remove old red blood cells from circulation. They only live about 120 days. The liver can pick up the slack, so the liver also does that. And then we just kind of already talked about that it is going to be controlled. It's going to listen to the pancreas. When there's a meal that we've eaten, then the pancreas releases insulin, and then the liver says, aye, pancreas, I'll do that job, and goes through glycogenesis. It's going to store that sugar, that glucose as glycogen for later use. And then when the glucose is gone, it's going to respond to glucagon released by the pancreas and go through glycogenolysis. And also, if there's not any sugar, then it can go through gluconeogenesis, and so it can make glucose from non-carbohydrate sources. And so lactate is one, so if you're exercising, your skeletal muscles are probably making lactate, and so it can rebuild it into glucose. If you're on that high-protein diet, like a lot of people are, the liver can also take amino acids, break off that amino group, will excrete that as urea, and make glucose. So gluconeogenesis, the beginning or birth of glucose from new sources, so from non-carbohydrate sources. It also makes plasma proteins, yes, so those albumins, those fibrinogens, which are those blood clotting proteins. Also, in the next unit, we're going to talk about cholesterol levels and how our fat is packaged and stored and how it moves throughout the body. The liver also, I don't know if you ever had to eat liver as a kid. I have to say I'm not a huge fan. My mom loved beef liver, which I just drenched it in ketchup and just choked it down. But one of the other jobs of the liver is it stores vitamins, particularly our fat-soluble vitamins A, D, E, K. The way you remember which ones are fat-soluble, it's awful, but it's great. All dogs eat kittens. And so A, D, E, K, and then vitamin B12 it's going to store for us, and it also stores iron. So it's actually a very healthy thing to eat, and it was cheap. There was 6 of us. So Mom cooked liver. And then I became an adult and never had to eat it again. It's awesome. Okay, so this is your money slide for the liver. We'll talk more about the metabolism of carbohydrates, lipids, and proteins in our nutrition chapter. So it has a major role in basically metabolism of all our nutrients. It's going to store vitamins and iron, also stores blood for us. We know that it filters and detoxifies blood. It'll also remove old red blood cells. And then 2, its digestive function is to produce and secrete bile. That's it for this section. Have a great day. See you soon.